So my name is Leo. Good morning again. I'm the sales director at Soileos. I'm delighted to be your host for today's session. We are pleased you've taken the time to join us for this session of Ask the Expert with Greg Patterson. Just before we get started, there are a few things that I want to go over with you. So this is an interactive session, so please feel free to submit your question in the chat box you will see in your screen or Q&A at any point during the conversation, okay? We will start with the pre-submitted questions first, followed by live questions submitted via the Q&A function that you see in your screen. We are incredibly fortunate to have with us today a leading expert in the field of soil analysis and fertility planning. So Mr. Greg Patterson. So Greg Patterson is one of the founding members of A&L Canada and is currently president and chief agronomist. So with an honors in agriculture degree from the University of Guelph, followed by over 35 years postgraduate experience as a professional agronomist, Greg works closely with all our agriculture clients to support the services of A&L provides. True agronomist at heart, you can find Greg supporting customers personally with in-depth agronomy and fertility training seminars for field horticultural and specialty crops. As well, this morning, we will be hearing from my esteemed colleague, Jason McNamee, so a Lucent Bio founder and GM of AGT Soileos. So Jason is a biogeologist and ecology expert with 20 years of experience in biotech research and development. So in his segment today, Jason will be focusing on the solutions Soileos can bring to your operation. So once again, thank you for being here with us. We will hand it off to Greg right now. So it's with you, Greg.
Thank you, Leo. Good morning, everyone. Thank you for inviting me to talk to you all today. I hope that we can cover off some of the questions or concerns that we might have in basic understanding of crop physiology and fertility and nutrient availability. It's a big question these days, and a lot of concerns over just all the different inputs we have, and especially when we're talking about soil health and how we move that forward and understanding how the plant uses this new technology. What we're going to talk about is how balanced fertility influences the plant microbiome, nutrient uptake, and the crop response in particular, because of the influence that nutrients have not only on the quality and yield of crops, but also the soil health that we'll refer to on a regular basis this morning. I want to first off start talking about the concept of soil nutrient availability. And the way we've looked at it in the past is that it's more of a single nutrient application when we look at trials that are performed by researchers. And that's really too linear approach in considering availability of nutrients in soil, because there's many other factors that have an influence on nutrient availability. Root absorption capacity is deeply a feedback mechanism controlled by the plant growth itself. So the plant has an influence on how much nutrient it's going to take up, and just basically the availability of nutrients to that plant is controlled by this plant and how it sets the stage for the yields that it's going to perform over season. When we look at this interaction, it's a lot to do with the different nutrients and the balance of these nutrients and availability of the plant in the soil. So it's more of a integrated approach, and that's where we're focused these days in understanding nutrient uptake and nutrient availability. Also, we're starting to understand that the plant also influences the interactions of microbial communities in the rhizosphere, which also have a big impact on how the plant takes up nutrients. So this bioavailability is very much a key component of how plants use nutrients. So in simple terms, it means the entire system is dependent on nutrient balance and the interaction between soil, plant, and the microbiome of the plant. So we have to start looking at it more holistically when we look at nutrient availability. When we talk about nutrient availability, we first of all look at the 17 essential elements and just how they're applied and the proper ratios that are required by the plant. So this balanced plant nutrition concept encompasses nutrient management, nutrient soil type, crop type, and stage of growth when we look at nutrient availability. And in that whole concept, we're looking at ensuring the proper ratios of essential elements that enhance the uptake of nutrients to the plant. And again, being able to supply these at certain stages of the plant's life cycle to make sure that the availability is there. So it doesn't stop just at NPK. And unfortunately, the focus has been too much on NPK and not really looking at the whole package. The first place we have to start with this whole concept with balanced plant nutrition is get over that love affair we have with nitrogen fertilizer. And that seems to be too much the focus. We need to look at balanced fertility, attention to all the essential elements in balance, and again at specific stages of growth, and understanding the soil's ability to supply these nutrients at any given time. And then the increased awareness of how we balance these soils, not just the soil nutrition, but the nutrition or our ability to provide a balanced nutrition system to the plant itself, and also focusing on building up soil health and the overall tilth of that soil. I refer to this particular study here because it's kind of outside what we see everywhere anymore. And this replicated study was done over 18 years, and it was looking at carbon sequestration, increasing soil organic matter, and just how we could actually do that to our standard practices. And their experience here was that increasing balanced fertility actually had a big impact on that overall increasing soil organic matter, soil structure, and again, this influence on soil health. So what they found in this study over the 18 years is applying a balanced fertility system increased the biomass and accumulation of nutrient uptake, and increase the organic residues that were turned back to the field. And again, there was a significant increase in soil organic matter where there was actually no organic residue other than the crop residue put in this field. But the balanced fertility supplied a better biomass that was turned back in the soil, and that had an influence on increasing organic matter. So it had a big increase on the organic matter, but also an increase on beneficial microbial populations in that soil. And again, what was noticed in this project is that phosphorus itself had a big influence on some of this increase in carbon sequestration and organic matter build, but also a positive association with the microbial population that was built up in these soils. So again, we're looking at it from a completely different aspect from what we've really seen happen in the last little while. So I want to refer to this diagram. I'm sure you've all seen this before, von Liebig's law of the minimum. It's all about which is the limiting factor in the field, and be able to identify that, and specifically not ignoring that all nutrients have a place, and the crop yield and quality is limited by the most limiting factor, which we need to identify. So in order to do that, we have to do a soil sample, analyze our soil, understand how to use that information. Our soil report is different than most soil reports out there because we've included a lot of research that we've done ourselves on understanding, first of all, soil variability and soil type differential. And this is an important factor because if you pick up fertility handbooks anywhere in North America, very seldom do they make a recommendation based on soil type differences. And we all know, simple concept, sands and clays are different. So we need to be looking at how we fertilize them differently to address that need. So our soil report gives you a recommendation of low, medium, good, or high based on what the soil type is first. And then from there, we make a recommendation. We also apply information on this report and help you understand it because it's about the recommendations that you can develop, not the ones that we generate from our computer system, because there's a lot of information we don't have at the laboratory that should be factored into your overall recommendation for growing that crop. So again, we rate the soil based on the soil type and give it a low, medium, good, or high rating on the different nutrients. We also provide percent saturation of the cations, so you can understand what is the predictable availability of these cations. And a lot of people don't believe in this concept. And I just want to clear the air a little bit here. We don't make recommendations based on percent saturation of the cations, but we put that information there so you, the agronomist or the grower, can ask the questions on what you would see as a potash or magnesium or calcium deficiency, so you can amend or change the recommendations we provide that, again, is not using all the information. The thing you have to understand is soils over the growing season, they basically release these nutrients, and that release of nutrients is based on a principle of soil dynamics called the coefficient of diffusion. And that's the ability of that soil, based on the water content in the soil and growing conditions in the environment, its ability to release the nutrients that it holds. Percent saturation is just a simplistic way of looking at that concept. So it gives us a better understanding that in dry weather conditions or good adequate moisture conditions, how much of these nutrients are actually going to be released. We also look at ratios of these nutrients, and the importance here to understand is how that influences the availability. I'll talk a little bit about this a little bit later. And when we talk about ratios, we're not talking about the ratios that Elbrick and Bray put forth in the 40s. We're talking about milliequivalents. And the reason why we break it down to milliequivalents is it does take into account soil type differences, and it does give us more information on how these nutrients play together. So we're saying don't guess soil test. It helps give a nutritional map of the farm so that you can make logical decisions of what you're going to do in crop planning. Removes a lot of the guesswork, and again, that forward planning and crop productivity. One of the biggest things you might have, or biggest challenge you might have in making recommendations in the field, is recommending a grower or even convincing yourself that it's probably not a good idea to put that particular crop in that field for this year, because there's things that have to be improved on before we invest the money in growing that crop. So it helps us with decision making on even what crop we need to grow. One of the biggest things when we talk about planning to deliver a fertility program is identifying these hidden hungers. And what I mean by that is we can't balance all soils, and we always will have some issue, either environmental factor or nutrient availability, that is going to limit that crop. And we need to identify what these hidden hungers are and implement programs of top dressing or foliar feeding or side dressing to identify some of these or improve on some of these hidden hungers before they actually cause yield loss. So that whole approach to forward planning of crop nutrient availability or delivery system is all about that forward planning. When we look at nutrient levels and we do our mapping programs at A&L, we basically try to paint the picture as many different ways as we can. For instance, here we have the cation exchange capacity of soils, looking at the variability of soil type from a sand to a clay across this field. Then we look at the different elements. In this case, we're looking at potassium in parts per million. And then we break it down into percent saturation levels to look at the predictable availability. But again, when you look at these two interpretations, they're very much two-dimensional. And it really does not take into account the soil type variability. So we have an indexing system where we actually take that two-dimensional image to a three-dimensional interpretation so we can understand even to a greater degree what is the variability and nutrient availability across that particular soil. When we talk about different soil types, back in the 40s when Elbrick and Bray and Baer were doing the research on fertility and fertility inputs, they were looking at specific soils that they were working on, and they really weren't accounting for variability in soil type. And unfortunately, it wasn't until 1974 that Fisher broke soil type down and took that information from Baer and Bray and Elbrick and basically said, okay, that's good to look at optimum levels of soil, but we now have to look at different soil types. And Fisher used cation exchange capacity to identify the different soil types and come up with categories of where those nutrients should be based on that early work. So basically all he did was identify the optimum levels across the different soil types and put them in some kind of category. And same thing with percent saturation, understanding in the different soil types, what concentration or saturation of these nutrients do we need to have a more predictable uptake by the plant. So if you understand this, the general rule of thumb is 3 to 5% of K, 10 to 20 of mag, 60 to 80% of calcium, and 10 to 20% of hydrogen. This is kind of looking at Elbrick and Bray and then put together as the general rule of thumb. The problem with this is that's not the way it is in the real world. We can't balance soils. We have to understand how the soil balance and plant needs might be variable across different soil types and design a fertility program that meets some of these inadequacies that we can't actually correct, and we have to correct it through the programs we put together. And you can see here CECs of 5, the sands, to have the right balance of potash and magnesium and calcium. One's going to come up short. So if we have the right K to magnesium ratio, we likely don't have enough calcium or enough hydrogen. So we're either applying more calcium or acidifying that system to make it work. CECs around 15, much easier to balance. Those soils are a little more easy to work with. But then again, as you get heavier clay soils, just the nature of the soil itself for these heavy clays usually have a higher component of calcium, and again, very difficult to balance. So we have to understand that and be able to design a fertility program through placement and other amendments to make the system work. So really understanding the dynamics of that soil and what its abilities are. We take it a little further when we look at that soil relationship than Elbrick ever did with his part per million relationships, as I said here. And we break it down into milliequivalents. In your sand soils, we like calcium to magnesium to be 3 to 1 in milliequivalents. Soils with silts to clay loams, 7 to 25 to 1. And then as we get heavier clays, a 6 to 1 relationship, but always maintaining a potash to magnesium ratio between 0.2 and 0.35. Now, this is impossible to do. There's no way you're going to make soils fit this perfectly. But if you understand these simple measurements and simple concepts, it allows us at a glance to look at these relationships and determine where our focus needs to be to make sure we can deliver the right nutrient package to the crops that we're growing. So this is just, again, a more simplified way of understanding what that relationship is to some of these major nutrients. When you look at CEC and texture, go from sands to clays, this kind of gives you a breakdown of what the CEC level would look like, so you can do a better job of understanding the soil that you're dealing with. We also really focus these days on soil health. Our soil health test is called Vitalis. It is a simple report that gives information to growers on what they need to do to improve the overall quality and health of the soil. The reason I mention this at this time is a lot of people think that soil health is all about reduced tillage, cover crops, green manuring, and it has a lot to do with it. But what actually makes it work is the nutrients we apply to that field and the crop we're growing, so that these plants can actually deliver the proper compounds or exudates to the microbial population to, one, attract them to the rhizosphere and then cultivate them. So this plant is going to cultivate this microbiome so it can fulfill the things it needs. So on this soil health test, it's a soil test with a lot of information in the background. We put charts and tables on this test to tell you as a grower or consultant, what the optimum ranges need to be. And these are things that we can do. These are things we can modify in the system. And we know based on the research we've done what nutrient, what combination of nutrients attracts the bacillus, the nitrogen fixers, the pseudomonas, the rhizobium. So we know in the background, based on this information, who's playing in the field and the things that we can simply do to encourage the better overall soil health and soil health level. We're looking at CO2 respiration, carbon, and again, our soil health index goes from 0 to 60. We want to get it over 40. Once you get over 40, you've got a very, very productive soil. And there's things we can do to make that happen. So this is kind of a simple recipe, if you will, to improve your overall soil quality. This starts with site-specific sampling your farm, or doing a soil audit, as we call it. And again, what we do for the grower is we give you a book of maps that basically paints a picture so visually you can understand what's going on. And I tell most people that farmers understand what's going on in their fields because they've worked on them for so long, but this is kind of a subconscious awareness. And what we try to do with our interpretation is bring that subconscious awareness to a conscious level by looking at just visual interpretations of that field and overall productivity. And our program's called TerraSite. And it's no different than taking a soil sample, but our interpretation of all the nutrients is in a map format like this that gives a color code indication. Again, green means go. The darker the green, the better the optimum level. And we just kind of color code it so that you could, at a glance, understand what those numbers actually mean. And a lot of times when I talk to growers, I tell them, just leap through your book of maps and stop at the map that looks like that field from the tractor seat. And if you identify that map, then we know specifically what we have to focus on. So again, it's just a color, it's a picture code so that we can really zone in on what's happening. What we need to understand is taking bulk samples in the field is not a good way of understanding your overall variability. This general fertility index shows you the type of variability that exists in this particular field. And the red again means poor, the greener it is, the better. And again, what we're trying to say here is one size does not fit all. So you really have to identify the variability and be able to address that, because if you're not defining the variability, you're not applying the nutrients properly. And we look at the different map interpretations that we say here to help you understand that. And again, we're looking at our general fertility index versus our soil health index. And you can see some similarities here where the fertility is very much influencing soil health. And you can break it down further than that and try to identify which nutrient is the key factor. In this field, potassium has a big factor or plays a big role in the overall soil health. in parts of that field, but it goes further than that. We also see that magnesium has some similarities to key soil health areas in that field. So we can start really zeroing in on nutrients and nutrient loads so that we can address these and not only get a better crop and better productivity with quality in the field, but improve the overall soil health of that field for sustainability in the future. And again, different nutrient interpretive maps here to kind of break it down to give a visual interpretation of how we see this particular nutrient, potassium, and its availability to that plant, with the potassium index being the most definitive in this case. And likewise with phosphorus, the same sort of thing. And we have one other key indicator with phosphorus. That's our phosphorus index, and that comes from understanding all the different phosphates and the saturation or the tie-up of phosphorus in soils due to iron and manganese and aluminum and calcium. So just a good interpretation of what's going on and being able to understand that. So I want to spend a few minutes talking about how we put a fertility program together, the things that we have to watch for. We look at Alder's chart. So we need to pay attention to when we apply something to a field, how it's impacting other nutrients. And you can see the complexity here of the interactions between the nutrients. You don't see sulfur here. Sulfur's a good guy. Most times we apply sulfur, it improves the uptake of all the other nutrients except for molybdenum. And you start getting aggressive with sulfur, we need to pay attention to that because molybdenum, although it's not deficient very often, it can become a problem. So things we have to look at, if we're using ammoniacal nitrogen in excess, it ties up potash, magnesium, and calcium. Nitrate in excess, phosphorus, sulfur, and iron, although it actually increases things like calcium and manganese availability. Phosphorus in excess ties up zinc, copper, iron, but also it increases manganese availability. So you see how these different nutrients play together. Potassium is unique. Potassium, we won't get into today, has a lot to do with yield and how the plant uses sunshine energy. But in excess, it ties up calcium, magnesium, boron, and copper. However, we have a relationship here, that antagonistic and synergistic relationship between potassium and boron, which I'll expand on here in a few minutes. And then magnesium in excess, potash, calcium, manganese. Calcium, another unique beast. In excess ties up most of the nutrients, but when it's not available, it also reduces the availability of all the nutrients because it plays a major role in how the plant functions at the root surface in the process of facilitated diffusion, which is how the root actually takes up nutrients. And we'll talk more about that in a few minutes here. So again, sulfur is a good guy. Most times it increases uptake. Boron, as I talked about, has a relationship with potassium, phosphorus, and calcium, and so on. So the point I'm trying to make here is when we put fertility programs together and you have somebody talk to you about using a particular nutrient, we need to understand that if we're going to use something we haven't been using, how is it going to play together with these other nutrients? How is it going to influence the availability? Or what are we doing now to reduce the availability of some nutrient that we're not paying attention to? And we need to understand that relationship. So you look at synergism, nutrients, it's not as straightforward as you'd like it to believe it is. Optimal supply of nitrogen ensures uptake of most of our nutrients, potassium, phosphorus, magnesium, iron, manganese, zinc, and so on. But again, we look at optimal levels of copper and boron improves nitrogen availability in the plant as well as molybdenum. So there's things we need to look at when we look at these synergies. Optimal levels of calcium and zinc improve uptake of phosphorus and potassium. Optimal levels of sulfur increase uptake of manganese and zinc. Optimal levels of manganese increase uptake of copper because these are transition elements. And then optimal levels of all nutrient increase nitrogen use efficiency. So once we balance these nutrients, we get better efficiencies out of our nitrogen dollar, and we can start looking at more efficient use of our fertilizer inputs. We look at the antagonisms, it's a little bit more difficult here. Excessive amounts of nitrogen reduce the uptake of phosphorus, potassium, iron, and almost all the secondary micronutrients like calcium, magnesium, iron, manganese, zinc, and copper. When we use excessive levels of phosphorus, we reduce uptake of a lot of the micronutrients, iron, manganese, zinc, copper. Excessive amounts of potassium reduce the uptake of magnesium to a greater extent, but lesser extent, calcium as well. Excessive amounts of calcium, as I said before, reduce the uptake of a lot of nutrients, in this case, iron. Our optimum levels of calcium also increase the availability to that facilitated diffusion and the activation of the mechanism at the root system to take that up. Excessive amounts of iron reduce zinc. Excessive amounts of zinc reduce manganese. And what we're talking about here is something we'll get into a little bit later. And these are all what we call the transition metals. And they will replace one another in the function of a plant. but they don't do what that element does. They just will interfere. So we can't apply one without the other, without paying attention to some of these antagonisms. We look at an inverse relationship between potassium and molybdenum. Low availability of potassium reduces the uptake of moly, therefore resulting in inefficient nitrogen utilization by the plant, which is something we have to pay attention to, and particularly at certain stages of growth where we can have an impact on some of the reproductive organs of the plant by causing this interaction. And a lot of times when we have potassium deficiency symptoms, the plant will show classic nitrogen deficiency symptoms. I'll talk a little about that in these next couple of slides. So when we look at interactions with nutrient like molybdenum uptake, the most, however, important is this reaction with sulfur. Sulfur does tie up molybdenum, and we're using more and more sulfur, particularly in Western Canada. So we have to pay attention to that in areas or in crops that are very specific and need molybdenum. Although it's not needed in large amounts, we need to make sure we have enough there to make it work. This is one that I really want to focus on because it's probably one of our biggest problems. And we look at using nitrogen, and we overuse nitrogen in most cases, but although nitrogen is fundamental to the production of proteins, plants deficient in potassium will not produce proteins regardless of how much nitrogen is available. And instead, incomplete proteins such as amino acids, asparagine, those types of things actually cause issues with nitrogen accumulation in the cell. And when that happens, we have issues with just overall quality. Starch synthesis is activated by potassium, and under inadequate potassium levels, the level of starch declines. And when it declines, we get water-soluble carbohydrates and nitrogen-based compounds that actually cause real problems with the quality of the crop we're trying to grow. So we get poor quality starch that's actually formed. We get soft, mushy cells. And a lot of times in these cells, it opens the plant up to more disease and insect attack because of these accumulation of these amino acids and this just poor quality of carbohydrates, particularly the attraction of sucking pests because of that nitrogen potassium imbalance. We look at soil levels and making this system work. We've identified that potassium to magnesium balance ideally is about 0.25 to 0.35 for most of our row crops. And the reason for that is the role that potash and magnesium in a relationship plays in the plant's ability to uptake proper levels of phosphorus. And we'll show you that in a few minutes here. If your K to mag is over 0.4, it indicates a magnesium availability problem, regardless of how much magnesium is in your soil. If it's below 0.15, it indicates low potassium availability, again, regardless of how many ppm is in the soil. And that's something important to pay attention to, because once you drop below a 0.15 in your K to magnesium ratio, your overall productivity in that field drops off dramatically. And we need to pay attention to that. So I said earlier that potassium plays a major role in the plant's ability to use sunshine and produce the photosynthase that it needs. And in a lot of crops that need high levels of potassium, like alfalfa or our root crops, it's hard to get potassium levels high enough unless our boron levels are high. And the reason for this is boron increases the ability for that plant to retain potassium or stops the basic efflux of potassium from the plant. So in these high potassium demand crops or high yielding crops, it's almost impossible to get enough potassium into the plant, regardless of how much is available, unless we have adequate levels of boron and boron availability. When we look at this relationship between nitrogen and potassium, we have to keep in mind that plants use potash to nitrogen at 1:1. So it basically has to combine with potash and nitrogen at the root surface, translocates up the xylem of the plant. Once it's up in the plant, the nitrates break off to produce the proteins and carbohydrates and things that we want to produce. And the potassium, as potassium malate, picks up the photosynthase, loads it in the phloem, and then translocates the photosynthase for redistribution through the plant and into the root and in the rhizosphere. So that relationship is important to understand. If we do not have enough potassium to empty the warehouse, if you will, nitrates are not used effectively by the plant. They accumulate in the plant, and this is where we have a problem with the quality of a high nitrate plant, causing more insect and disease pressure and just basically reducing our yields. So that efficiency of nitrate use by the plant is very much a potassium nitrate relationship, but also we can include boron in here. And this is just a slide showing the way boron has an impact in the overall construction of the phloem. And on the left-hand side, we have adequate boron levels in the plant, and you can see the well-structured phloem. That is the delivery mechanism for the photosynthase throughout the plant. And on the right-hand side, we have low boron levels, and you see a very unorganized phloem system, which has a big impact on how well this plant can translocate photosynthates around the plant. So this is kind of the connection. We look at potassium and boron and nitrogen and how they play together and how important that is to understand. As I said, sulfur increases uptake of most nutrients. When we look at a soil report and we look at nutrient availability, the cations play a major role. But a simple thing here to look at is how much hydrogen is available in your soil. And the reason for understanding that is sulfur plays a major role in helping the plant have enough hydrogen in the rhizosphere for uptake of very essential elements like nitrates, phosphates, sulfates in the early season, because hydrogen plays a role as a companion ion for the uptake and translocation of something like phosphorus across the root membrane. And if we don't have enough hydrogen in the soil solution, the process of active transport of phosphorus into the plant is reduced, and we just get very low energy produced in early season. So I use sulfur and this information not just for sulfur as a nutrient, but also in understanding how sulfur plays a role in helping with the acidification of the zone around the plant to give us better early season uptake. So just something I want to take a minute to help everybody understand how the plant actually functions. The plant actually has to produce energy to drive everything. And that energy comes from conversion of inorganic phosphorus to a form of phosphorus the plant stores as ADP, and the first process in the production of the carbohydrates in the plant. So this process is how all plants basically work in early season. The plant wants to take up salt, and the salt of choice is potassium, and it sticks that salt inside the plant root system to start that osmotic pull of water and nutrients through that passive process. And you can see here that process takes energy, and that energy comes from that active transport where we use energy to actually pick up the potassium and move it into the plant. That energy comes from the use of inorganic phosphorus, the phosphorus we apply in the spring, or the phosphorus that's available in the soil. You can see that there's 2 hydrogen with every phosphorus taken up. That's the companion ions that help phosphorus be taken up. And the conversion of inorganic phosphorus needs an enzyme that's synthesized with potassium in the plant to basically make that process work. It also needs magnesium, which erupts from the mitochondria in the early stages, to make that bridge and convert that ADP to ATP, which in actual fact is magnesium ATP that the plant stores as energy and uses in the whole process. So understanding that relationship between potash and magnesium and phosphorus is really a real understanding of how the plant basically gets started and how it develops the energy to drive the whole process throughout the life cycle of the plant. And I talk about this because it's important to understand that in understanding nutrient balance, how important these relationships are. This is a simple picture of that process of how phosphate is taken up by the plant. You can see the hydrogen moving in through that portal in the roots at the root surface to actually help facilitate the uptake of phosphorus. So hydrogen is actually pumped out of the plant through this other portal here. You see the hydrogen moving up that portal. And again, that takes energy. And energy comes from the breakdown of ATP back to ATP to release that hydrogen and pump it out. So the plant is trying to acidify the zone around the root system naturally. And anything we can do to help improve on that efficiency with a little bit of acidulation in our fertilizer band at early spring soils, it is also a benefit to increase that overall energy the plant needs to get started. So we need to understand this chemistry a little bit just to understand what the plant's trying to do naturally. And the fact if we put a fertility program together, we don't want anything close to that small little root system that is too alkaline in nature, because we don't want to disrupt this slight acidulation that has to take place. I want to talk about boron for a few minutes and just talk about how important it is to the plant and kind of the misunderstanding we have about boron. Most people think it's very toxic to plants and we can't be putting it on. Our research has shown that for most of our soils, we're very deficient in boron for which causes a number of problems. Most people think that 1 part per million of boron is sufficient, and we're going to show you here that that's not the case. If our CECs are below 18, 1 to 1.5 part per million might do the job, but as you get in the heavier clay soils above 18, we're going to have 2.5 to 3 part per million just to make boron available to the plant. And soils right across Canada, that's not the case. We're lucky if we have 1 part per million. And so we're very much dealing with soils that are boron deficient. And I'm going to show you here how much of an impact that has on crop production. So we know that there's antagonism and synergies with boron. The more potassium we put down, the more we reduce boron availability from soils, so we get more tie-up. The more we increase boron levels in the plant, the more we retain potassium in the plant. Most people think that boron is influenced mainly by pH, but pH has a very slight impact on boron availability. It's more about the clay content of soils because boron has a definite affinity to clay. And the higher the clay content, the more boron is bound by the soil and the harder it is for the plant to actually get boron out of the soil. So that's why we have that sliding scale of boron when we look at the EC. So this is just a chart showing that pH has an impact on it, availability, but again, not nearly as much as clay. So the pecking order here is for influence on boron availability to plants. First of all, your clay content. Organic matter plays a role in high organic soils. It also binds up boron and it makes it less available. Then pH, then moisture, then temperature, and so on. And then we can talk about excessive levels of potassium also in soils, reducing availability of boron. And this is just a chart out of a piece of literature that shows that impact to CEC on the boron availability. Our chart looks something like this in boron availability across different CEC ranges. This comes from our own research, and we see dramatic influence to boron applications, even though many people said boron's not available. Is toxic, but it's not available if we have less than 1 part per million in a lot of our soils. And putting a lot of boron too close to seed, yes, can cause a problem. But we have so many boron choices these days, different products that we can use to make boron more available without causing some of that issue with toxicity. We look at, this happens to be tomatoes. I'm sure there's not too many tomato growers in Western Canada, but again, we see quality aspects with potassium levels, and these levels of boron have to be much higher than we're used to using. We see with increased application of boron, we see less cracking, just better overall quality, better fruit firmness, better shelf life, and again, much better yields when we look at that. And the problem here is everybody's of the opinion that 20 part per million in a tomato is sufficient. But we're finding that to get the quality of tomato we're looking for in the yields, we're going to need up close to 40 to 60 ppm in petioles, which most people would tell you is toxic. So we're looking for this information here. Again, either foliar applied or ground applied has a big impact on overall potassium content in the crop. So again, stopping that efflux of potassium from these high potassium using crops is very much a part of boron nutrition. And again, here, just overall conclusions on boron availability and that consistently treatment of boron improved overall quality. And you see here by this chart that we've got to get close to 60 part per million of boron application consistently to get the levels where we want for the quality we're looking for. And in this case, we're looking at boron levels in excess of 60 to 80 part per million to get the quality of the uptake of potassium that we want in this particular crop. And likewise, in any high potash demanding crop, alfalfa, even canola and sunflowers, we need more boron to get the potash uptake to get the yields we're looking for. And that 20 part per million that a lot of literature has is old information, and it comes from databases that are basically generally average databases where there has not been a lot of attention to boron nutrition and the impact it has on overall quality. The other thing that we're involved in soil health and microbial activity, and we're tracking how boron has an influence on overall CO2 respiration. And just what can we do to make that happen? We're also seeing crops that have high yields. We automatically see higher levels of boron in the plant tissue as the plant actually reaches out and sequesters a microbe that helps it attract or take up boron that would otherwise be less available. And from this research, we're looking at someplace around 3 kilograms per hectare, or 3 pounds per acre, basically, per season applied to these soils to increase this microbial activity. So what we're saying here is that a lot of this soil health we're looking to and a lot of the overall microbial activity that's major influence on how this plant's going to respond and the sustainability in our system is a lot to do with these microbes and how we can stimulate these populations to be more aggressive and more active. And it has a lot to do with boron nutrition and these microbes. So that's a mouthful, but it's something I had to put in there because of the research we're doing on a continual basis on crops. We're seeing that we're just not using enough boron, and everybody has this idea that it's toxic and is dangerous to use. And our yield limitations from just overall physiology, the maintenance of the phloem, the the uptake and retention of potassium in the plant is a lot to do with boron. Our cell structure, calcium and boron make up the cell wall. Just so many things that we just have not paid enough attention to. So let's look at micronutrients in general. The first place I like to start is talking about the quality of the micronutrient you use. A lot of people buy micronutrients based on the price. Some products are a lot cheaper than others. But if you're using a micronutrient, I don't care if it's zinc, manganese, or what it might be, availability is key. Either bioavailability or water solubility has a big impact on how these things perform. And this is just a chart showing you that, the difference between zinc sulfates and oxysulfates and different forms of zinc, what one is the cost, the other is the solubility, the availability of that particular micronutrient. And using a micronutrient that has no solubility or availability is just a worthless attempt. I mean, we look at total zinc in products and solubility ratings. I mean, you get below 50% solubility in something like zinc, and you might as well left the stuff alone because it's not giving you any crop response. This is not our work. This is from Mortvet, some work that was done by researchers for the fertilizer industry, just showing what solubility looks like. This is going from 0 to 20 pounds of a zinc product, an application. This happens to be corn, but you can see this particular product, which has virtually no water solubility. It was worthless. Just at any level, it did nothing for that crop. Whereas you have something that in this case is 66% water soluble within 22 days, similar to the other slide, you can see the impact of the different rates and the plant's ability to take up that particular zinc. So you look at products, don't let price be a determining factor on how you're putting this together. Buyer beware. Look at what is the impact. If you're putting a product on, there's things you should see. Not necessarily will you see an increase in tissue uptake because it gets diluted, but look at the overall visual effects. And I'll show you some visual effects here in another crop here in a few minutes to help you understand that when we look at nutrition and how they play together, some of the underlying impacts these nutrients have on the overall take-up. So looking at the micronutrients, zinc, manganese, iron, copper, I already mentioned this once. These are transition metals. They do compete with one another, and they can take a role in masking the other nutrients if you're used out of balance. So we have to really pay attention to how we put these together and how they play together. To have adequate zinc levels using our soil test, which is a hydrochloric extraction for zinc, I want to be greater than 5 part per million to have ideal zinc availability. In some crops that are really zinc sensitive, I try to build soils to get to that level. Look at manganese, I want that north of 33 part per million, make it available. Look at iron, 25 part per million. And copper, particularly in the West, because of the amount of cereals and grains we grow, that should be 3 part per million. So we look at these transition metals. Again, they will compete with one another. And to have adequate levels in soils, they need to be above the levels indicated here. Again, we look at relationship between something like manganese and iron. We need 1 part manganese to 2 parts iron to make them both play together. Some crops like potatoes, where we need 40 to 60 ppm of manganese to control something like scab and just give us the emergence we want. That would mean we'd have to be greater than 50 part per million, which would be too high for something like copper, unless we're higher in copper. So we really have to look at the way these things play together, or we can cause more problems than we solve. So identifying zinc deficiency, that's what it looks like in corn, and that wide banding of the yellow. And the reason I use this slide is the experts for years and years had this confused with manganese, and I'll show you that relationship here in a few minutes. But when you look at global zinc issues, we see that there's a lot of zinc deficiency in the soils worldwide. And you see the zinc deficiency in North America in the green, that they say that's medium zinc deficiency. It's actually higher than that. We see that in a lot of areas, in crop producing areas in Canada particularly, we have some fairly serious zinc deficiency. Again, we want it above 5 for adequate availability, or we need to be able to apply it and make sure we have enough for the crop. I look at critical levels. Again, I want to be in that 5 range or above the 4.9. So 3 to 4.9 is medium, and we want to be above those levels to make it work. So we look at micronutrient availability. A granular zinc fertilizer should have water solubility greater than 50%. You're going to hear something later today, I think, about bioavailability, which hasn't really been measured that well, but The factor has a lot to do with microbial activity and just how these things perform and become available to that crop and uptake. And it's really important to understand that total zinc is not enough to determine zinc availability to a crop. We need to be managing that and making sure that we have a good understanding of that. So look at how zinc responds to crop performance. We know that zinc is important for root growth. It's important for all meristematic growth in the plant, and we need good root growth early. I mean, it has a lot to do with geotropism in the plant, and I just want to walk through that a little bit and help you understand that. So with zinc, it forms indolic acetic acid in the plant, which is responsible for all that meristematic growth, and it helps with elongation of the root. And in doing that, what happens is zinc, through the gravitational pull, will be pulled, the indolic acetic acid pulled to the base of that root. And when that happens, it comes in contact with cytokinins, and the cytokinin actually basically causes a callousing when these two come in contact. And that callousing stops the growth of the bottom part of the root here. And we still have elongation of the top part of the root. And that gives us that root's ability to go deeper in the soil. So root penetration of soil has a lot to do with early zinc availability, and your overall root growth has a lot to do with that availability. And that's why it's so important to have zinc in a planter mix or available to that plant in early season. We also know that zinc can be toxic at high levels, but plants have a built-in ability to produce indolic acetic acid oxidase, which actually controls that concentration. And that is very much regulated by boron again. So when we have boron deficiency, we have excessive indolic acetic acid oxidase, which completely cannibalizes the indolic acetic acid and starts to cause an impact with how these nutrients play together. So when that happens, we have a real concern with when we put planter mixes together with zinc of not having boron with it, because this relationship is very important how these work together. So we look at zinc availability and root growth. We now want to look at how the crop comes out of the ground and early emergence, and the apical or hypocotyl, when that starts to emerge. There's no cytokinins in this part of the plant, so we don't get the callousing, and we actually get accelerated growth with indolic acetic acid being pulled to the base of that emerging hypocotyl, and it turns the plant sprout up and out of the soil. So big impact on the plant's ability to emerge properly. And this is just showing you seed corn placed or seeds placed basically upside down, and the plant's ability through indolic acetic acid to turn that and right that emerging sprout up and in the right direction, and the root system going down basically at about a 25-degree angle down. So zinc availability, we put planter mixes together. We need to pay attention to that zinc and phosphorus relationship. We need to balance that. So in good zinc with medium phosphate, for every 10 pounds of phosphorus, you need a pound of zinc. In soils that have high zinc levels, you can get away with 20 to 1, but for the most part, we like putting about to keep that 10 to 1 in mind when we put together a fertility program, so that in using phosphorus, we don't further influence zinc deficiency. One thing I'll talk about is a piece of physiology that plays a major role in a lot of crops that we don't pay enough attention to, and that's that zinc builds cells. Again, that whole concept of cell differentiation and meristematic growth. With adequate levels of zinc in the soil, when the plant's developing cell structures and cells, the higher the indolic acetic acid, the more cells that are developed. This is simply, could be a leaf tissue, could be an apple, could be a potato root, whatever. The cell or the plant on the left has low levels of indolic acetic acid from zinc, so it does not produce as many cells when the plant's being differentiated, either leaf growth or fruit growth or whatever it might be. The one on the right is higher concentration of zinc, higher concentration of indolic acetic acid, and larger concentration of cell differentiation and cell development. Every one of these cells are held together by a calcium-phosphorus bond. Every one of these cells are surrounded with calcium. So zinc in early season increases the plant's ability to differentiate and grow bigger cells, and this happens to be in leaf, in anything, but also it creates a calcium sink. So if we do not have enough zinc in the plant early, we do not develop a proper differentiation at flowering time to create a cell structure that gives us better bulk density and just better overall cell structure. So we look at that relationship. Here's a simple diagram. We've got here a picture of two different applications of zinc. This is a potato crop early. Both crops had 5 pounds of actual zinc in the row, but that was not enough to compete with the phosphate we put down. So we come in and foliar fed the one on the right, another early application of zinc to increase the leaf area to give us the type of leaf development we wanted. That just shows you the difference. So same thing with that cell differentiation. It has a lot to do with leaf growth and leaf size. So leaf area is impacted by early zinc as well, which gives us that better leaf, which has a lot to do, in this case, of this crop, in the amount of tuber initiation that's developed because of that leaf to shoot ratio that's so important. And so you see here the difference in those two. And again, root growth and quality, the amount of tuber initiation and the amount of increased tuber initiation from that bigger leaf size. So some of these underlying things you don't see that's going on in a plant when we apply something like zinc that you don't look for. So when you're looking at zinc and zinc needs, don't always look at the tissue sample. Look for some of these physiological changes in the plant you're growing to make sure that, in this case, zinc is doing what it's supposed to do. This is just looking at corn, seed-treated zinc and untreated. This is onion, and this is an important one here. You see that this flag in the leaf is a typical zinc deficiency. And where this plays a role, and it goes back to this whole calcium thing, and I use this demonstration on onions to demonstrate this. If we have low levels of zinc, we don't get the leaves developed we want, and the leaf has a lot to do with how the bulb forms. So we need early season zinc to get us the cell differentiation, to get the leaf differentiation, to create the bulb that gives us the quality onion. If we don't have that, we get soft rot, we get a soft neck here, and bacteria infection actually causes more soft rot. And that's the type of thing we see in the onion in storage with low zinc levels. And this just shows you a rating in disease and zinc in parts per million in the affected plant versus the ones that aren't affected, and just kind of where that magic number is in this particular crop, all to do with zinc. And we follow that back even further with this onion bulb, and you can see here the zinc that has the onion has higher concentration of zinc, has almost 1,000 part per million more calcium in that bulb. So that goes back to what I talked about in the plant's ability to actually absorb calcium that's applied because of the larger number of cells differentiated and just basically creating that source or that sink for the calcium to actually end up in. This is the same sort of thing in apples from a foliar feeding program versus just application of calcium. And again, zinc here had a bigger influence on calcium uptake in this particular crop than the actual foliar calcium did. We look at grain and we look at efficient soils and grain growth. Again, when we plant a crop that has been grown on a field with low zinc levels, just the overall vigor and growth of the grain we plant is going to be impacted. You can see here grain with 12 milligrams per kilogram in the grain that we planted versus 35, a substantial difference in overall growth and performance of that grain as well. Talk a bit here about manganese and manganese deficiency. Seeing more and more, again, we're not going to get into why with the use of glyphosate, but it's something we need to pay attention to because a lot of these crops are very dependent on manganese, and it's not really noticed, or we don't often pay attention to it. And manganese is one that's hard to build in soils. It takes about 2 years to actually start seeing a build from a ground application of manganese. And we need to combine a good granular program to build these levels plus a proactive foliar program when we identify these deficiencies in fields, showing manganese deficiency in wheat and the types of things we see in strips and the types of impact it has. Being able to identify this and differentiate it between other nutrient deficiencies is important. Tissue samples obviously can give us that information, but we should be actually using a soil sample to see where those levels are and be a little more proactive than waiting for a tissue sample to bring that information to us. This is manganese deficiency of corn. The reason I put this up is for the longest time in Ontario, that manganese and zinc deficiency symptoms were confused. There's manganese on the left and zinc deficiency on the right. Again, zinc deficiency being a broader band of chlorosis than the manganese, which is more striping. So again, we want to be greater than 33 part per million on our soil test. 20 to 30 for crops that aren't really not responsive to manganese will be sufficient. Still some hidden hunger there, but for the most part, I want to be over 33 part per million for things like grains and legumes. And again, this shows you the kind of the range we want to be in. I think you've all heard enough about what manganese does in the plant and the synthesis of chlorophyll, germination and maturity. We talked a little bit about increases things like phosphate and calcium availability, And again, we need to be able to identify this. But one more important issue with manganese issues is in germination and emergence. If we have adequate manganese levels in our grain and in our soils, we get better emergence. And the reason for that goes back to how this thing comes out of the ground, how that hypocotyl actually emerges through the soil has a lot to do with manganese levels in the emerging plant. When we have low manganese levels, what happens is as this crop comes out of the ground, it slows down because of that lower manganese level. So your hypercotyl just basically stops growing. And something like rhizoctonia is an opportunistic disease. And anytime a plant has a hard time emerging, it gives more opportunity for that rhizoctonia to infect the plant. So again, I can relate a lot of rhizoctonia issues in crops due to poor manganese availabilities in early season. This is just a marker again showing that in a research project here, we had all kinds of manganese in the first stage here. You see good linear growth. Take manganese away, it slows down, add it back, it takes off again. So just showing you here that adding manganese back over the time period of this manganese deficient trial, that it had an impact on our main axis growth in early season. So just again trying to demonstrate how important manganese for early growth. So we know manganese problems are high pH soils, basically lower manganese. Again, high organic matter soils have an issue. And just like all the other micronutrients, cool, wet spring conditions have an impact on things like manganese and zinc availability. And then again, poor biological activity can have an impact on any nutrient availability to this crop that we're growing. We look at manganese nutrition, soil applied versus foliar fed. We want to build soils to 33 part per million. And if you're starting down at 8 to 10 part per million, which a lot of soils are anymore because of the issue with manganese levels dropping, it's going to take you a couple of years to get that above that. And it's going to take about 45 pounds of actual manganese to go from an 8 to a 33 part per million. And that takes time for it to happen. And we usually tell people that it's going to take 24 months to see your first application even start showing up on a soil test. So as we build soils, we start broadcasting manganese to build soils. You have to have a proactive foliar program to address the crop's needs until the soil is sufficient enough to supply that. A band application of 3 to 5 pounds can override some of that. So that's another effective way of getting it in. But again, identifying where you have manganese deficiency and putting a proactive program in place is really the key to get that done. We know that we have to broadcast it to improve it. In most of our soils, we suggest a 15 pounds per acre application rate for the first year, and then again the second year, and then wait a year and resample your field to see if you need to have another application to get over that 30 part per million. And the reason for that is manganese is expensive, and you want to make sure you understand how much you need. And again, that band application, the 3 to 5 pounds can override some of this, but it takes time. This is just kind of a diagram showing how manganese actually uptake looks. First year, we see at 15 pounds, we see really nothing happening in the soil. We will see some crop response, but not significant. And then we see after the second year, we start seeing that build start to take place as the manganese starts to work in. And it's really not till the third year that we see a significant rise in soil test manganese levels, as it takes about 24 months for the soil to actually absorb it and start to release it. Talk about copper deficiency, huge issue with copper deficiency right across Canada. We don't pay enough attention to it, mainly because people are afraid of it. It is toxic, and it's using copper sulfates hard on sprayers, so people stay away from it. And we have a lot of varieties in Western Canada wheat that, even though they're copper deficient, are very sensitive to copper application, so people stay away from it. So it's a concern. The safest place to put copper is in the soil. It takes, again, a little while for it to build. We need copper for grain crops greater than 3 part per million of a hydrochloric extraction. So that's where we want to work to. And applying a copper sulfate to the soil can get us there fairly quickly. But again, we have to make sure that we're not causing crop damage when we apply that. When we look at some of the facts around copper, it has a lot to do with chlorophyll formation. It's a lot to do with sugar translocation within plants. So a lot of things that we don't see happening with copper. Promotes better seed production, essential for a lot of enzyme activities that we don't recognize. Again, that the biosynthesis of lignin, the quality of the straw strength, all that has a lot to do with copper. This is just showing copper deficiency in wheat. It's pretty pronounced. Again, that flagging of leaf tip, head development, copper with adequate copper and in healthy versus crop deficient heads. And again, just looking at some of the things we don't really focus on with copper because we don't see it. Protein formation in the plant, electron transport, carbohydrate metabolism, nitrogen fixation, sugar translocation, and a lot of crops, skin finish and quality has a lot to do with pollination, particularly in grains. These are things that are very subtle that we don't really recognize. Mineral soils versus high organic matter soils, high pH or high organic matter soils have a lot of issues with tying up copper. Again, that 3 to 7 ppm is where we want to be on mineral soils. But on organic soils, I want to be up to 15 part per million to make sure I have enough available copper in the crops I'm growing, because it's usually horticultural crops like onions and carrots where skin finish has a lot to do with copper levels. That's just some of the things that cause copper deficiencies or interactions in copper availability. So again, very subtle things that we don't pay attention enough to, and most people don't identify with copper. You do in the West more because you know the significance of copper deficiency in wheat, but in the rest of Canada really goes unnoticed. We look at correcting the problem. Again, copper sulfate's the easiest way to do that. We've got trials in grain and in other crops where we've put on up to 15 pounds of actual broadcast copper sulfate, and that seems to fix the problem and last for a number of years. But again, copper sulfate's not nice to work with, can damage sprayers, and growers just don't like messing with it. But it is a proactive and a good treatment for correcting copper deficiencies that's long lasting. And looking at ergot and sclerotinia wheat, the impact copper has on it. Again, there's lots of research out there that shows that and identifies that, and just the gloom emergence with proper copper levels versus insufficient copper, and the increased flower abortion and disease in grain. Powdery mildew, another real issue we have on wheat, and the impact that copper has on it. So a lot of things that we see and we deal with on a regular basis with something like copper. So in summary, balanced fertility provides consistent yields and quality, while at the same time builds a more sustainable ecological environment for growing our crops. So we really need to look at soil health and balanced fertility's impact on soil health, and not everything, but also this whole issue on carbon sequestralization, as I mentioned earlier, is really impacted by overall balanced fertility. So with that, I'm going to turn it back over to you, Leo. I hope we didn't lose everybody, but I'm open for questions.
Thank you, Greg. Very insightful session. I have a few questions for you, if you don't mind, please.
Looks like we lost a lot of people.
No, I don't think so. People are very engaging. So
Okay.
we have first one come from Alberta.
Yes.
It says some labs show only nitrate nitrogen, some labs show only ammonium nitrogen, some labs show both. I guess that wasn't very the topic here, but he has some he wants to learn more about that. So if you can explain more about this situation, I think it will be very clarifying for him.
There was some research done about 20 years ago in Saskatchewan, I believe, on analyzing soils for ammoniacal nitrogen and using that in the calculation of nitrate recommendations for crops. We get the odd grower from the West that still wants us to do that. We will do ammoniacal nitrogen and report it. It'll cost more to do that. I don't use that in my calculation. I rather use nitrate nitrogen and estimated nitrogen release from organic matter to come up with and yield goals, obviously, for nitrogen recommendation. But again, the reason for some labs doing it, and most of those labs that are doing it, again, are based out of the Saskatchewan area that do it just as a matter of fact because of that early research. We don't get very many requests for it anymore because we have nitrate nitrogen that we're looking at, we have yield predictions, and we have estimated nitrogen release that gives us a pretty good understanding of nitrogen is available. My only comment I'd like to comment here on is that nitrate nitrogen's a bit of an elusive number. Mother Nature, again, determines when we have nitrogen released, and there's lots of cases where we have hot, dry summers and we don't get nitrates released. We don't get the numbers we'd expect in our fall soil samples, and growers scratch their head because they don't believe the numbers. And then later in the season or next spring, we got all kinds of nitrates. So we really don't understand the nitrate test as much as we believe. We do the best we can with the numbers we have, and that's why it's good to use ammoniacal nitrogen as another factor, or estimated nitrogen release as a factor, or our Savita nitrogen burst test for understanding nitrogen, because it's a difficult one to get a real solid handle on just because of the way microbial activity has an impact on how it's released from the products and organic matter in soils that make it available to the plant. And that can really confuse things, and it's a hard one to get a handle on. So anything we can use to get a better understanding of nitrate availability in soils to make that recommendation is why we have all these different tools we're working at. But yeah, it's elusive, I guess. We do nitrate tests across Canada and the states. Every province and every state has a different way of calculating nitrogen needs from a nitrate test. That tells us we all don't really understand what we're doing. So it's a real question. It's a real question, Mark. It's a good question. You have good reason to ask that question.
Yeah.
I hope
Okay.
I
Well,
answered it.
yes, you did. So thank you, Greg. A second one come from Manitoba. Thanks, Chad, for submit this question. So are we seeing more tie-up of micros due to different ratios, such as P to zinc ratio, N to S ratio?
Yes. And that's why I spent the time talking about the antagonism and synergisms. Too often, a grower will sit in a coffee shop and his neighbor will say, well, I use this zinc product or this manganese product. I had amazing things happen. And he goes out and buys the same product and puts it in his field, and he gets a negative impact. And that's because he didn't identify it, or they didn't identify, what was the real antagonism or what was the real deficiency. So a lot of times, and that's why I talk about these relationships and the fact that in the case of micronutrients, they're transition elements. A lot of times, unless we really understand why we're using it, we do understand that we have either a soil test level that's deficient or tissue samples are telling us we're deficient. We've identified those hidden hungers. We can do things that actually create problems for ourselves. So it's really important to be able to identify why we're using something. We're not just using a shotgun approach. We're using it because we've identified through historical information or through soil samples or through tissue samples that deficiency is actually there. So understanding how they play together is really important.
Yes, thanks, Greg. That this now, Greg, this isn't actually a question; it's a comment during the session from Dennis, and I just want to share with you to get your insight on that. Dennis mentioned there is a lot of very good information that is presented very well in this presentation. So I guess that's for you. So the roadblock, if any, with producers is the economic ROI on providing some of the product and technologies. They will work with it up to a break-even point or a point they stop getting paid for their efforts and cash outlay. Perhaps a longer view will be taken by some.
That's a mouthful. And that's exactly what happens in the industry. A lot of research is done based on a certain allocation of funds. You might get results in the first couple of years, but by the time the project's supposed to complete in 3 years, we really don't have a conclusion. One of the biggest problems we have in research that's done on all nutrients is we're still looking at a linear approach. So to get an ROI on something when you're not paying attention to the whole balanced fertility system might not prove out. Because if you start looking at using copper on wheat and your biggest deficiency in that field is potassium, you're not going to win by putting down $50 worth of copper. I mean, it's just not going to work because you have bigger fish to fry. So we really have to understand that, as I said in earlier stages, that we have to take more of a holistic approach to what we're looking at, because looking at nutrient needs in a plant on a single element basis will oftentimes give you a negative response or will show you no ROI. So when you're doing a response to let's say you're looking at zinc or manganese or copper, you need to make sure basically before you even start looking at that research that all the other nutrients are satisfied. So you've got a balanced nutrition program before you set out to start looking at, if I increase more copper use or increase more manganese use, what is the return on investment, if that makes sense? So a lot of our research, and that's why I started out with that slide, a lot of our research is based on this linear approach, and too often research findings will find that there's actually no return on investment by using that micronutrient or that product. But they haven't looked at that. They've looked at it on a linear approach to it, and they haven't gotten rid of all the other possible nutrient interactions that would basically show no results. So we do research, we put a plot together, we use our general fertility index, and we index all the results based on that so that we take background fertility out as a determining factor. And then we can start seeing the real response to what we're applying. So we're not always in a situation can we fix all the problems, all the underlying imbalances, but if we index the imbalances, when we apply a nutrient and do our best at trying to fix them, you get a better understanding of what is the return, if that makes sense.
Yes, yes, Jason, please.
Thanks, Greg. I guess my comment there would be that would assume that we're doing proper soil testing and that we have the information from all of the fields so that we can then make those decisions.
Correct. Well, why would we do research without we do a lot of research, and my technicians will give me grief all the time because when I tell them we're going to do a research project, I want soil tests, I want complete soil tests, I want ammoniacal tests, I want nitrate tests, I want tissue tests every week, along with a nitrate test. I might not use all the information, but I can't go back and get it. If I have a null hypothesis or poor results, I can't go back and say, well, what caused that? Because I don't have all that information. So answer your question is, why would you do research on a crop if you didn't have a soil test?
And how does that then translate to the farm? Because you're doing research and you have a certain budget for that, but growers are typically limited on their budget, and like you mentioned earlier, a lot of people are just doing the composite sampling. How do they justify that cost for the deeper level of sampling?
Okay, the site-specific sampling we recommend in Eastern Canada is because we have smaller fields of work, one every 2.5 acres. In Western Canada, we talk about one every 5 acres. And we started doing site-specific sampling. I had a lot of growers say to me, well, how can I justify that? I say, well, okay, you may never sample that field that way again. But if you don't have a good understanding and do what I call a soil audit of the variability you're dealing with, how do you identify what's working? If you do a site-specific sample, in future years, you can go out and take a sample from what you've classified as a low area or a high area to see if your fertility program is moving you forward or backwards. So you're not going to sample that field that way again because you're just going to select these what I call these reference points. And the farmer says that makes sense, and the economics makes sense. I'll do it once and at least I have a good understanding. I've never had a grower in 35 years come back and say that they use that. They've always wanted to go back and sample the whole field because they find the information and understanding that variability is so valuable in everything they do and how they buy fertilizer, how much fertilizer they apply. Is variable rate technology paying me? Why am I having that response? Because now I drive across that field and I see that spot. Now I have a map that tells me why it's there. How do I focus on it? So again, bringing that subconscious awareness to a conscious level with that, what I call that soil audit, helps the grower deal with the farm base he has to see what kind of investment he needs to put in that farm to grow the crop he needs to grow. And where does he focus his dollars? Does he use copper? No, he's got a phosphate problem. So fix that first. So it's about the economics of using the information properly and be able to get a good understanding of what is going on in the field, but having a reference point that I can get back to year after year after year and get a good understanding. One thing we showed this 2 years ago at the Ag in Motion in Saskatchewan, we soil sampled the field and we did our maps and we showed farmers the variability they have in nitrates across that field. And they never thought of that. They do a bulk sample for nitrates and they get a test. And some years it doesn't agree with what they expect, but they got the information from a bulk soil. And if they had site-specific sample of the field at one time, and they saw where nitrates would be higher or lower based on that soil profile, they can start using those site-specific samples as a better indicator of nitrate levels before they start making their nitrogen prescriptions. So it's just the more information we have, the more detail we have, the better we are at applying the dollars in the right place.
It reminds me of the famous Peter Drucker School of Management saying, which is you cannot manage what you do not measure.
Right.
That's right. He says that.
Yeah.
Great insight, Jason. Thanks, Greg. Greg, we have another one come from Alberta. Eduardo, I think we know him. So considering the last dry years in Western Canada, can you please talk a little bit about soil health and water holding capacity?
Yes. Again, the work we've done on soil health and the measurements we've had, we very much have seen fluctuations in even our soil health index in dry years. We know that when we have dry soil conditions, your microbial activity slows down. And those beneficial microbes that are doing so much for the crop basically go dormant, and they're not helping release nutrients, uptake nutrients, suppress disease. They're just not active. Now we know as we increase our soil health, we increase some of these Pseudomonas and Rhizobium, these organisms secrete a polysaccharide that actually aggregate soils together, and these polysaccharides also retain more moisture. So as your soil health improves, your microbial activity improves, these organisms actually secrete a compound or help the plant secrete a compound that retains better soil moisture. So your healthier fields, the higher your soil health index, even though your microbial activity in a dry year will still diminish somewhat, over time will hold more moisture. If that, I don't know if that is what you're looking for, Eduardo, but did I clarify that good enough?
I understand. So that's okay. Thank you, Greg. Very insightful. So Eduardo is saying here, perfect. So I guess you answered the question. So thank you for that. So Greg, very insightful. We appreciate your time. Now we have in the presentation from Jason. So Jason will share with you guys a little bit about Soileos and how we can support your operation. So thanks, Greg, and Jason is with you.
Thank you.
Yeah, thank you very much, Greg. And I also want to say to our team here, thank goodness we're recording this, because I'm going to have to go back over Greg's presentation a bunch of times to pull all the information out of there. It was pretty speedy, but it was really great. And the other thing I'd want to say thank you to everybody who's still here. We're an hour and a half in, and we've only lost a couple of folks. So I'm very grateful for the opportunity to present to you. But I won't keep you too long. I made sure it was short because I knew Greg would go long. Let's see, share 1 sec. Okay. Yeah, I'm going to talk a little bit about who we are as a company, talk a little bit about who I am as a person being able to have this conversation with you, a little bit about our technology, and I'll touch on some of the points that Greg also went over with respect to current suite of technologies in this space. I'll just share my screen here. Oh, we can see that okay?
Yes.
Great. Thank you. So our company is a combination of our good friends AGT Foods, and the technology comes from Lucent Biosciences, where we developed this new development in the core fertilizer technology, which we call Soileos. And Soileos is the product that we manufacture in beautiful Rosetown, Saskatchewan. Okay, there we go. This is who I am. My name is Jason McNamee. You can see I'm a prairie boy through and through, originally born in the Balgonie area, played hockey at Notre Dame. I have the privilege to serve as the Chief Operating Officer at Lucent Biosciences, where I'm also a founder, and I'm also privileged to serve as the General Manager and a founder at Soileos. The other thing is, what really drives me, and this is, I think, drives a lot of our company, is I have these two young men here who were the rationale for me leaving my geotechnical environmental engineering career to start off on this path to improve outcomes. So we know there's some problems, and these are some of the problems that drive us at Lucent Biosciences and Soileos. I like to think of things on a 10,000 year scale, because 10,000 years ago is when humans decided to stop chasing animals around, and we became civilized. This was civilization started when we started becoming farmers. And we can see that over the last about 9,850 years, the human population hasn't really grown very significantly. It was very steady. And then same with the CO2 in the atmosphere until about 150 years ago, where we experienced exponential growth, where we had 800 million people on the planet that lived to an average age of about 32, whereas now we have 8 billion people that live to an average age of 70. And we can see also the same curve over in CO2. I don't like to really think of it as climate change is our main problem, but change is the issue that really drives us. And we are trying to decrease the uncertainty as we move into the future. And so those are problems, those are big real world problems that drive us. But our farm customers also face a lot of problems in uncertainties. Like we have these tariffs, sometimes they're on, sometimes they're off, but that does impact the certainty for the farmer to go forward in his planning. And there's also uncertainty in the market. So how do we decrease that uncertainty and increase the positive outcome for the grower? And that's what we are really working on. And we have done an awful lot of work to be able to be here and to say to the folks here that we've developed a new innovation in the core of fertilizer that typically is working better than competitive solutions, improving ROI. In addition, our product improves soil health. So to Greg's discussion, we improve the soil microbiome, as our product is a food source for soil microbes. and we also sequester carbon through this process. So the advantage with our product is defined in the title of the patent, which is composition, systems, and methods for delivery of an element in response to biological demand. If there's no biological demand, there's no delivery of our products. So when Greg was talking, he was talking about solubility of elements. Our products are water insoluble, and this is one of the first products to roll out in this new category, where our product is a food source for soil microbes rather than a salt that dissolves when it hits water. And the advantage with that is our product releases crop nutrition when your crop requires it optimally. That is when the soil is warm and moist. So our product is about 75% carbon, 10% potassium, 5% sulfur, and somewhere around 10% of a micronutrient. The percentage of the micronutrient is dependent on the nutrient itself, so that it does change a little bit. But this carbon-based product is what we have that really differentiates us from the rest of the technologies that are currently available. And so one of the advantages with our product versus the current suite of technologies is that nutrients are only released due to biological demand. And then if we think about what is the crop removal rate from the crops that we typically are planting here in Western Canada, well, we can see this from this chart, maybe a little hard to see here, but the crop removal rate for most of our crops is less than 0.1 pounds of actual micronutrient per acre. And it is also, or maybe 0.2. And so what we have been able to do as a company, we have done thousands of tissue and soil tests that show that at the end of a season, there is residual zinc and improved levels in the tissue after 1 year. So we have a product that can last more than 1 year, provided we're soil testing and we're confident that that is the case. So then typically, I like to ask where to use these products. So over the last number of years, we've done several hundred trials across Western Canada. And in addition, we have done hundreds of trials in research greenhouses and figured out, where our product provides the best R for the end user. And, it's not really a surprise, but our answers are very similar to what it says exactly on the Saskatchewan Agriculture website, is that we find that under very high or very low organic matter conditions, we get a significant ROI for the end user. We find that in coarse grained soils, like sandier soils that are typically lighter in color, we have better results and improve our ROI. And we also find that as we get into higher pH conditions, that our product works where conventional products don't work as well. And so when we think about where, when, and why we would use a product like ours, we would look at our soil tests first to tell us where the right place is. But in absence of a soil test, we would look at some of our more marginal lands and say, that is where we probably are going to have a better outcome in terms of yield, production, and return on your investment. So I'll go through this quickly. This competitive technologies that we're used to seeing in the industry. First one is oxides, which Greg showed. They're not water soluble and in fact not bioavailable. However, we do use them because they're convenient and they can be micronized and added to fertilizer and that sort of thing. But we have to remember that in order for us to use an oxide, the zinc and oxygen bond, or iron and oxygen bond, or whichever micronutrient it would be, that bond must be hydrolyzed by microbes or by acids or low pH. So it does sort of like an oxide does sort of satisfy the requirements of a soil test, but it is the least bioavailable of the technologies that are available. I'll talk a little bit about sulfates. Sulfates are very common in this space. They are both water soluble and bioavailable, but they do come with their own suite of issues. And that is very typical of most of the salt-based fertilizers. One is timing. So if you're applying your product and it's a cold, wet spring, we will still get dissolution of the granules, and that can result if the biology isn't there to take advantage of the ions that are in solution, we typically will get tie-up or we will get leaching. Tie-up, again, is one of the issues with water-soluble product because it has to move through mass transport through the soil, and there's competition for positively charged cations in the soil through clay, because all clay minerals are negatively charged, and we again can get tie-up through clay. Or you heard Greg talk about a relationship between phosphorus and other micronutrients, which makes them less bioavailable. Another thing about sulfates is they're typically fairly concentrated, 20 to 35% concentrate. And that means we're typically applying about 1 pound of actual zinc per acre. And that means that on a 35%, you're getting very few granules per lineal drill foot, which makes the access for each individual plant it's more difficult to get that zinc moving around in the soil. And then if we talk about salt index, the salt index in a sulfate is in the high 80s, where a product like ours is below 30. And then the last technology that you're used to is a chelate, chelate's a neat word. It means claw. It's a Greek word, means claw, and it basically is a molecule that holds a zinc or an iron or one of these large cations so that it doesn't tie up in the soil. It doesn't get tied up to another positively charged ion, which is great. And they absolutely work. The problem with the issues that come up with them is that when you use a chelate molecule to get to a plant, you're also adding a chelate molecule to the plant, which the plant was never looking for in the first place. And there's evidence to show that those chelates decrease plant performance while still delivering the ion. The other thing with chelates is they are almost all liquid applications. So these are foliar applications. And we find that foliar applications, often they are missed, or by the time we see a problem at a foliar application timing, we may fix the problem from a superficial viewpoint, but it's already too late to fix the problem that was already created by the issue. And then the last thing again is distribution. Is sometimes water-based or foliar-based can be difficult to do based on the timing of and equipment that you're using. And the last thing I want to touch on with our product, I had a way longer presentation, but I knew Greg's was going to be long. So I also wanted to touch on is that our carbon-based fertilizer also is able to retain moisture. And we ran this replicated study with the Prairie Agricultural Machinery Institute out of Humboldt. And what we see is that our product on its own, under freeze-thaw tests, you don't get any clumping at all. Our product, when blended with typical salt-based fertilizers, did a pulse blend and a wheat blend for this, and we'd be happy to share those results with you, is that our product mixed with those blends decreased the amount of clumping, which helps us be more confident that when we go in the spring to pull our fertilizer out of the bin, that we're not going to have to use what Nicole likes to call the persuader, which is the hammer just to get the fertilizer out. So with that, I'll give everybody their time back, and if there's any questions, we're happy to answer them at any time for you. I can't hear you, Leo.
Hello? Sorry. Yeah. Thank you, Jason. Thank you one more time. So Nicole is sharing with everyone some important links to support Jason's presentation in the chat. So feel free to check it out when you guys have a moment. I guess we don't have any specific question for Soileos. Just before we transition to our closing, I would like to thank everyone for participating of this webinar. So it's been almost 2 hours and we still have the most of the people here with us. I really appreciate your interest in learning more about importance of micronutrients, as well as how Soileos can support you in your micronutrient plans. I personally had some notes in the 3 important things that I understand we can share validate the conversation today is that micronutrients, they are just as essential as macros in a successful fertility plan and balanced nutrition. So while macros they fuel growth, understand micros drive the critical biological functions that ensure plant resilience and optimal performance. Another thing that is important, bioavailability is key. So only nutrients that are available for plant uptake will positively influence soil health, plant health, yield potential, and most important, ROI. And a very important note I have is that Soileos supports your fertility program. So we deliver a complete solution that combines essential micros with high bioavailability, ensuring nothing is left behind for your crops. So I think that is my notes for today. I really want to thank you guys again. So thank you for joining us. We hope this session provided you with very valuable knowledge that will optimize your fertility plans. Once again, we appreciate you taking the time to join us today and allow us sharing with you the role that innovative solutions like Soileos can have in your crop plans. If you have any specific question, just reach out to our sales team. So we have Eduardo in Alberta, Danny and Jason in Saskatchewan. We have Kern in Manitoba and myself in Ontario. So we will be happy to discuss with you on how Soileos can be a valuable addition to your fertility program. Another important message, I will be sharing in a recording of this webinar and additional resource via email in the coming days. So until then, please feel free to check out the Soileos blog, our website, soileos.com, and our podcast, the Back 40 Broadcast. And also follow us on social media. So at Soileos for more agronomy content and updates. I want to also thank you, our market team is always here supporting us. So Nicole, Devin, and Isabel helping us putting this together. Thank you so much. So thanks everyone once again. I hope you have an awesome Friday and I'll see you next time. Bye.